All right, this video is about nucleic acids. This diagram should look familiar to you. It is a three-dimensional diagram of a DNA molecule in its double helix shape. Nucleic acids include DNA and RNA. We're going to start with DNA first. And nucleic acids are made of the elements carbon, hydrogen, oxygen, nitrogen, and phosphorus. DNA stands for deoxyribonucleic acid. So sometimes students forget what a nucleic acid is because they forget that that's what the N and the A stand for in DNA and RNA. So if you ask them, oh, what's DNA? They know that. And if you say, what's a nucleic acid? They sometimes don't really remember that DNA is a nucleic acid. DNA is the hereditary material passed down from one generation to the next during reproduction. It is a polymer made of a monomer of nucleotides. So the monomer is called a nucleotide. Here is a diagram of the nucleotide. Each nucleotide has 3 parts. It has one 5-carbon sugar called deoxyribose. It has one phosphate group right here, bonded to the sugar, deoxyribose, and a nitrogen base. Sometimes you'll see nitrogenous base, sometimes you'll see nitrogen base. Whichever is easier for you, that's fine. And the backbone of the DNA is made up of alternating sugar and phosphate. The sugar is the hexagon, the pentagon. So it goes phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And you see it on both sides. Now the sugar-phosphate backbone just gives DNA its structure. It's not really informational. The DNA, you may know, is referred to as the blueprint of life. It is actually the nitrogen base pairing and the sequence of that actually determine all of our traits, not the sugar and the phosphate. Not that the sugar and phosphate aren't important. They're very important in the structure of DNA, but they're not informational. If you think about DNA as a recipe, think of the words, just like with these letters, as the bases, and the paper and the thread that holds it together and the binding of the book would be like the sugar and the phosphate. So there are 4 bases: adenine, thymine, guanine, and cytosine. Adenine always pairs with thymine, and cytosine always pairs with guanine. You need to memorize those base pairings. Now, this, of course, is flattened out. It looks kind of like a ladder, and the sides of the ladder here, and these would be the rungs of the ladder, the part where your feet would go. But in reality, it's twisted in shape, in a 3-dimensional shape called a double helix. Now, this base pairing is referred to, or has a vocabulary word that's associated with it, which is complementary. It is the most important vocabulary term when you're talking about DNA. And it is actually the complementary nature, the fact that, first of all, that DNA is double-stranded, and the fact that A and T only are attracted to each other and G and C, that make DNA able to be copied before it's passed on to the next generation. So again, if you're talking about a DNA molecule, you always want to talk about A going with T and G going with C, and that those 2 bases would be complementary to each other. It's a very important vocabulary word to always use when you're writing about DNA. So this is just another diagram showing you the complementary nature of DNA, and it's also showing you what one nucleotide would be. One nucleotide is one sugar, one phosphate, and one nitrogen base. So if you want to look at a molecule and count how many nucleotides it would be, you just count the bases. So 1, 2, 3, 4, 5, 6, 7, 8. There would be 8 individual nucleotides would have to come together to form this piece of DNA. DNA is much longer than this. They're just showing you a piece that could be shown on the screen and fit. So again, this whole thing here is the sugar-phosphate backbone. And we have nitrogen bases, we have sugar and phosphate. And one nucleotide would be one sugar, one phosphate, and one nitrogen base. And they are held together by hydrogen bonds, the bases that is, and you can tell that because there is a dotted line between them. You are not responsible this year for knowing the difference between a purine and a pyrimidine, but in AP biology you will be, or in college biology you will be. So adenine and guanine are purines, and cytosine and thymine are pyrimidines, but you will not be tested or quizzed on that this year. So RNA, which is referred to as ribonucleic acid, is directly involved in protein synthesis. Contains ribose instead of deoxyribose, which means it has a different sugar. And it contains uracil instead of thymine. So 3 out of the 4 bases are the same, but one of them is different. It is also single-stranded instead of double-stranded. So if you're looking at a diagram on a test or quiz, and you see that it is double-stranded, well, you know that's a DNA molecule. If it's single-stranded, you can't be sure that it's RNA because DNA molecules come apart when they're being replicated or copied, for those of you who are not familiar with the term replication. So what you really need to look for is whether there is thymine or uracil. If you see uracil, you are 100% sure that you are looking at RNA. If you see thymine or T's in the diagram, you know for sure you are looking at DNA. Now, another difference between DNA and RNA is that DNA can never leave the nucleus. RNA does leave the nucleus. It's made in the nucleus, but leaves and goes into the cytoplasm and finds ribosomes. So DNA is like that reference material that you're not allowed to check out of the media center because they're afraid that somebody else might need it and it wouldn't be there. And so think of DNA as like when you have 46 chromosomes because you are human. And think of each chromosome as like a recipe book with very many recipes in it. A gene is one recipe, and it is a recipe for making proteins. And we're going to do an entire unit on protein synthesis that will show you how the cell uses the code in a DNA molecule to build an RNA and then a protein. But DNA, because RNA exists, DNA doesn't need to leave the nucleus. RNA will make a copy of one small piece of this DNA molecule, one gene at a time, and take that recipe to make one protein to the ribosomes. And ribosomes are where proteins are made. So that is another difference between DNA and RNA. So what is it about DNA that makes it the perfect molecule to copy, to then be able to pass down to future generations? Well, it's double-stranded, and those 2 strands are complementary. And we're going to get into some more details about this later in a later unit, but the DNA molecule can come apart and a new DNA strand can be built on each of the old strands in a complementary fashion. So it is the double-stranded and complementary nature of DNA that makes it easy to copy.